Hi, I'm Kalaiyarasi Ragunathan, a master's student from Molecular Medicine program. I chose to do my master thesis on the impact of acetyltransferases in B cell development under the supervision of Dr. Valentin Oxenich. Here are the two major outlines of this project. Firstly, I aim to investigate the impact of acetyltransferases GCN5 and PICAF in class switch recombination of B cells. As the second part of this research, I decided to study the acetyltransferases acting in the early B cell stages. The cells are the structural units of organisms, which contains the package of DNA that carries the genetic information. Due to the lengthy structure of DNA, the cells tangle up DNA with proteins known as histones and form a compact structure called chromatin. Since all the cells in the body obtain the same genetic sequence, how do they function distinctly? The epigenetics is the answer to this question. The epigenetics is the chromatin modification that regulates the gene expression of the cells without altering the DNA sequences. This includes histone modification, DNA methylation, and non-coding RNA modifications. During these alterations, DNA, RNA, and histones are coupled with small chemical tags which eventually change the gene expression. To date, a long list of histone modifications has been identified. In this project, my main target is histone acetylation. Generally, the histone proteins gain positive charge through arginine and lysine residues in their structure, which attracts negatively charged DNA strands and form compact nucleosomes. During the acetylation, negatively charged acetyl group from the donor molecule, acetyl-CoA, will be transferred to lysine residues on histones, which subsequently relaxes the chromatin structure. Further, the histone acetylation is strongly correlated with the transcriptional activation of genes. The key enzymes mediate acetylation are called histone acetyltransferases. Based on their sequence similarity and structural domains, they are classified into several families. The GCN5 and PCAF are enzymes from GCN5-related N-acetyltransferases family, which is briefly called GNAT family. Both are homologous proteins, sharing nearly 86% sequence similarity. The GCN5 and PCAF contain three distinct domains: catalytic H80 domain, which mediates acetylation reaction; carboxy-terminal bromodomain that helps the attachment with DNA; and N-terminal PCAF domain that facilitates the attachment of P300 and CREB binding proteins. Further, these proteins have been identified to be involved in several cellular functions, such as embryogenesis, neural development, T cell activation, DNA damage response. The B cells are vital components of immune system, and their developmental process includes several intricate mechanisms. One of the crucial stages of the B cells is class switch recombination. As the name implies, the class switch recombination allows switching the classes of antibodies by changing their constant regions of the heavy chains. Generally, the naive B cells carry immunoglobulin M and immunoglobulin D in their surface. After interacting with antigen, the cells undergo isotype switching in order to produce antibodies with distinct effector functions and the same antigen specificity. The class switch recombination is driven by the enzyme activation-induced cytidine deaminase, AID in simple words. It creates the breaks in S region of donor and acceptor gene segments. These breaks will be eventually resolved by double-strand break repair mechanisms. In the end, a recombined heavy chain genomic segment is formed. An intervening region will be excised as circular DNA. And according to a study by Lee and his colleagues, primary B cells treated with GCN5 and PCAF specific inhibitor possess significant reduction in isotype switching of immunoglobulin G1 and immunoglobulin G3. In other words, lack of GCN5 and PCAF activity leads to reduction in class switching. Based on these findings, I hypothesize that GCN5 and PCAF are playing essential role in class switch recombination. To assess this hypothesis, I use CH12F3 cells, which are capable of switching from immunoglobulin M to immunoglobulin A with high frequency after in vitro stimulation. I used two distinct strategies to generate GCN5 and PCAF lacking cells. Initially, I enzymatically arrested GCN5 and PCAF in wild-type CH12F3 cells using their specific inhibitor, CpTH2. Then the cells were treated with in vitro stimulants, interleukin-4, anti-CD40, and transforming growth factor beta for 72 hours, and immunoglobulin A levels were analyzed using flow cytometry. In another method, single knockout and double knockout cell lines were generated using CRISPR-Cas9 gene editing technique. Then the cells were treated with in vitro stimulants and immunoglobulin A levels were analyzed in the cell lines. Interestingly, the cells treated with CpTH2 showed nearly four-fold reduction in isotype switching when compared to untreated control cells and solvent control. In this experiment, AID deficient cells, which cannot maintain this class switch recombination, were used as negative controls. However, in this strategy, GCN5 and PCAF were simultaneously attenuated by their specific inhibitor. Therefore, these results only reflect the impact of combined inactivation of GCN5 and PCAF. Then, to assess their individual contribution in class switch recombination, I used single and double knockout cell lines generated by CRISPR-Cas9 technique by Dr. Carroll in our group. I found consistent outcomes from both the methods. The double knockout cell lines show a drastic drop in class switching of immunoglobulin A when compared to wild type and single knockout cells. Further, I saw a substantial reduction in class switching in GCN5 null cells when compared to PCAF deficient cells. After considering both the methods, I found that my hypothesis is true. Both the GCN5 and PCAF are essential for class switch recombination. In addition, the impact caused by GCN5 inactivation is comparatively higher than the PCAF deficiency. However, the combined inactivation of GCN5 and PCAF can lead to even more severe consequences than absence of either gene alone. Further, this finding suggests the redundant functions of GCN5 and PCAF in B cell development, mainly in class switch recombination. The next step, I was determined to find the reasons for the reduction of class switch recombination in the cells lacking both the GCN5 and PCAF. A successful isotype switching depends on several events such as AID production, AID recruitment, germline transcript production, double-strand break repair process, and cell proliferation rate. In 1996, Hodgkin and his colleagues showed that the isotype switching capacity of a cell is profoundly correlated with their proliferation rate. Taken together, I hypothesized that the reduction of class switch recombination observed in GCN5 and PCAF deficient cells is due to the impaired cell proliferation rate. To test this hypothesis, cell proliferation rate was assessed in different cell lines at different time points 24, 48, 72, and 96 hours, and there was no significant change in cell proliferation rate observed in any of the cell line when compared to the wild type control. These results prove that my hypothesis is wrong and raise the conclusion that the GCN5 and PCAF are dispensable for proliferation of CH12F3 cells. Well, the two important steps in isotype switching are generation and repair of double-strand breaks at the S regions in the immunoglobulin heavy chain locus, and it's quite clear that the isotype switching can be severely impaired due to deficient double-strand break repair. Therefore, I hypothesized that the class switch recombination reduction observed in the GCN5 and PCAF deficient cells is due to impaired double-strand break repair pathway. In order to check this hypothesis, I treated the cells with the drug etoposide, which induces the double-strand break in the cells. The etoposide blocks the function of the topoisomerases by obstructing their ligase activity. The topoisomerases are major enzymes involved in resolving of supercoiled DNA. The figure on the left down corner shows the function of the drug. The etoposide molecules are shown in yellow. This intervenes into the nicks created by topoisomerases and blocks the resealing of the broken ends and facilitates double-strand breaks. At the end, double-strand lesions will be resolved by double-strand break repair pathways. Through this experiment, I found that the etoposides treatment in distinct genotype did not cause any significant change in the cells. All the cells were equally sensitive to the adipocyte, so my hypothesis was wrong, and this shows GCN5 and PCAF are dispensable for the double-strand break repair in CH12F3 cells. Now, turning to the second part of my research, in which I determined to investigate the acetyltransferases acting in the early stage of B cells, especially pre-B cells. Several studies have proved that the acetylation on the histone 3 lysine 9 residue is a hallmark of a transcriptionally active gene. Acetylation of this specific lysine residue is mediated by GCN5 and PCAF enzymes. I found that lack of GCN5 and PCAF in the mature B cell line CH12F3 completely abolishes the acetylation of lysine 9 residue of histone 3. The western plot of CH12F3 cell line on the left-hand corner shows the lack of lysine 9 acetylation in GCN5 and PCAF double deficient cell line. The results from the VABL cell lines are counterintuitive. The VABL cells, which correspond to the pre-B cells, show no change in acetylation of histone 3 lysine 9 after inactivation of GCN5 and PCAF genes. Based on this, I hypothesized that the acetylation of this specific lysine residue in VABL cell line depends on more acetyltransferases than GCN5 and PCAF. To test this hypothesis, I used set of inhibitors on the cells to enzymatically arrest the acetyltransferases. Initially, I treated the cells with distinct concentration of GCN5 PCAF specific inhibitor CpTH2. The CpTH2 molecules build bridge interactions between lysine residues and ultimately block the reaction mediated by the acetyltransferases. The wild-type CH12F3 cells treated with CpTH2 possessed reduction of lysine 9 acetylation. At the same time, wild-type and GCN5 PCAF double-deficient VABL cell lines were observed without any change in acetylation after CpTH2 inhibitory treatment. As the next step, I treated the cells with global acetylation inhibitor anacardic acid, which is a non-specific acetyltransferase inhibitor. The anacardic acid interacts with the acetyltransferases by an unknown molecular mechanism and eventually reduces the global acetylation in the cells. As a result of inhibitory treatment, all the cell lines showed a substantial reduction in acetylation of histone 3 lysine 9, including the VABL cells. It means that some unknown acetyltransferases mediates histone 3 lysine 9 acetylation in VABL cells, and it can be blocked by anacardic acid. I also enzymatically attenuated P300 and Krebs binding protein with their specific inhibitor PU139. The inhibitor blocks the P300 and Krebs binding protein by interacting with their conserved cysteine residue in catalytic H80 domain. The CH12F3 cells treated with PU139 showed a gradual reduction in histone 3 lysine 9 acetylation. But this specific lysine 9 acetylation in VABL cell lines was not affected by the PU139 treatment. These experimental results indicate that additional so far unknown acetyltransferase modifies histone 3 lysine 9 in VABL cells. This acetyltransferase is distinct from GCN5, PCAF, P300 and Krebs binding protein. As a summary of the findings from this study, firstly, I found that GCN5 and PCAF are essential for class switch recombination in CH12F3 cells. On the other hand, these proteins are not involved in cell proliferation and DNA repair. Further, I found that acetylation of histone 3 lysine 9 residue in early B cells is mediated by new unknown acetyltransferase, which is distinct from GCN5, PCAF, P300, and Krebs binding protein. Overall, the proteins GCN5 and PCAF have a complex role in B cell development. In future perspective, I would suggest that AID production, AID recruitment and germline transcript production levels should be analyzed to elucidate the precise role of GCN5 and PCAF in class switch recombination. Further, an extensive study, including an array of acetyltransferase inhibitors, should be carried out in order to identify the enzyme acting in early B cells. Finally, I would thank my supervisor, Dr. Valentin Oxenich, for his guidance and continuous support throughout my dissertation period. And I thank my co-supervisor, Dr. Carol Beck, for teaching me practical and theoretical work. Further, I thank my team members, Sergio, Amin, Shindong, and Oxenich Research Group. Thank you.